Good morning to everybody. I may make a request first for everybody to stay muted while we do this. To the fellow members of the Asian community, WAPI members, IACS members, and the IACS/IAWW leadership team led by Lalita Upala. So on behalf of Washington of Indian origin, it really gives me a tremendous joy to welcome everybody to this inaugural Zoom webinar on COVID-19, which is a hot topic right now in the health and safety of all of us. So this is geared towards community education and awareness, and hopefully this sets the stage for many future WAPI and IACS collaborative events. I believe Dr. Amish Dave is already working on a couple more later this year. And I do want to thank, first of all, Dr. Amish Dave for spearheading this amazing activity for community education, and also putting the panel together with support from IACS team. Want to also thank Ms. Lalita Upala to get excited about doing this activity for IACS and IAWW community. And I think we are in for an amazing 2 hours of COVID-19 scientific presentations and panel discussions and question and answers. So let's go for it. Thank you everybody for being here.
Well, thank you so much, Sanjeev. Again, we have a fantastic lineup of speakers today to talk on the various facets of the COVID pandemic, and they're going to all give us scientific presentations. A few housekeeping points before we start. To all the audience, please keep yourself muted. We will take questions at the very end. So if you want to send us your questions on the chat box, we have a moderator collecting the questions, and we will take them all to our panel members at the end. So please keep your questions on the chat box. And again, we want to get the speakers going first. So to kickstart our webinar, I want to first call upon Dr. Helen Shu. Dr. Helen Shu is an associate professor in the Department of Medicine, Allergy and Infectious Disease at the University of Washington. She earned her MD at the Duke University and master's in public health at the University of Washington. Her research focus has been on preventive interventions against influenza, RSV, and emerging respiratory viruses, including the SARS-CoV-2. She's one of the principal investigators of the Seattle flu study. So as you may know, the Seattle flu study is a multi-institutional community-wide virus surveillance program, which was started in 2018. And Dr. Shu and her team spearheaded to recognize that COVID-19 was getting transmitted in the community in the US for the very first time. She also heads the UW vaccination trials and all the emerging treatments in treating the COVID illness. So we are in for a treat. Please welcome Dr. Helen Shu.
Thank you so much. Let me see if I can pronounce it correctly. Dr. Trikudanathan.
You got it.
Okay, I'm pretty good at South Asian names. And Dr. Dave for inviting me today to speak to you. It is really such a pleasure to be here with you on a Saturday morning. So thanks to all of you for coming and listening to the talks today. I was tasked with talking a little bit about vaccine development for coronavirus. And so I'm going to go ahead and get started. Feel free to send questions my way. Let's see if I can. Okay, great. So here are my disclosures. Let's see if I can. Here we go. Okay. So in my talk, I'll be going, maybe, you know what, could I have someone else handle slide advancing? Because it seems like I have quite a bit of delay on my side. Would that be okay? Okay, I'm going to go ahead and get started. So in my talk, I wanted to talk briefly about sort of the pathway towards coronavirus vaccine development, and then talk specifically about how we got to SARS-CoV-2 vaccine development, and then go over the current vaccines that have been have received FDA emergency use. And then talk about some of the issues that are currently ongoing in the world of vaccines, including what we're doing for pregnant women and children, the effect on asymptomatic transmission, the role of the new variants, and then some unanswered questions in the field. Next slide, please. So the top emerging pathogens likely to cause severe outbreaks in the near future. This was actually something put together by the World Health Organization several years ago before the COVID pandemic. And when they came up with their list of the pathogens that were most likely to cause another pandemic, you can see that coronavirus is right there on the list. So I think what this tells you is that the world knew, the WHO knew, the NIH knew, everybody knew that a coronavirus was one of the possibilities for the cause of the next pandemic. And so a lot of the work to prepare for this pandemic has really been done in the last several decades based on some basic structural biology work that's been used to address these challenges around coronavirus, therapeutics, and vaccine developments. Next slide. So this is a picture of a bat. The bats are thought to be the source of a lot of emerging respiratory viruses. The idea is that there is a zoonotic source, so an animal source to many of these pathogens, and then they travel over to an intermediate host and then over to humans. So part of the work to be done in surveillance and preparation for the next pandemic is to develop platforms in parts of the world where you're likely to see another pandemic virus emerge. Next slide. So when we look at coronavirus, specifically, coronavirus is actually a lot of different types of viruses all coming together. There are the human coronaviruses, which actually cause quite a bit of common cold symptoms. And last year in Seattle, we found that it caused 15 to 30% of common colds in the population. We also know that SARS-CoV is is a beta coronavirus. So when you look at where it sits relative to the other ones, it sits very close to SARS-CoV-1, but not in the same category as MERS or the human coronaviruses on this phylogenetic tree. Next slide. When we think about how human coronaviruses came over from animals to humans, what you can see here is that most of them started in a bat or a rodent. They all then have an intermediate host. For MERS, for example, it was a camel, and then it jumps over to humans. So we expect that the next pandemic virus is also going to emerge this way. Next slide. So based on, I'm just going to give you just a very little bit of background on anagenic structure of coronavirus, because it helps understand the vaccines that are being developed for coronavirus. So we know that from studies of SARS coronavirus 1, the one that caused all the SARS epidemics about 2 decades ago now, and MERS coronavirus, that the spike protein, the S protein on the surface of the virus, is really the ideal target for a vaccine. When we look at SARS coronavirus 1 and then the current SARS coronavirus 2, this particular surface protein, the spike protein, interacts with a receptor on the human cell called ACE2. And so when you have antibodies that target this spike, what you're doing is you're blocking the spike from attaching to the human host cell and initiating the cascade that causes infection. Next slide. So this is a close-up of that particular, of coronavirus, and coronavirus has what it looks like a crown around it, and that's why it's called coronavirus. And then this is a close-up, the structural characterization of the spike protein. The spike protein is metastable, so it means that sometimes it's in a certain position, and then it flips as it attaches to the host cell. And that is something that's common to spike for SARS-CoV-2, but it was also found for many, many other respiratory viruses. And I think what this tells you is that there's a common way that viruses are attaching to host cells. And so a lot of these pathways, all of the basic science work over the last 2 decades allowed us to get to a vaccine very quickly, because it allowed us to be able to use this information that we've gained from all these other viruses to get to vaccine development for SARS-CoV-2. Next slide.
लगता है बहुत पहले से शुरू हो गया.
This is also showing you the spike protein. Basically, it occurs in this metastable form. And then what happens is then it flips over as it attaches. And then something that we talk about a lot in vaccine development is RBD, which is the receptor binding domain. It's the specific piece of spike that binds to the human cell. Next slide. Okay, so this is courtesy of Barney Graham at the NIH, who developed the mRNA Mrna Vaccine. He made these slides, and I use them all the time, and they're great. And what you can see here is that there's the spike on the left, and then the human cell is on the right. Next slide. And then you see how these two are attaching, and this is how the virus gets into your body, gets into the cells. Next slide. What happens when you have antibodies? So if you give a vaccine that has this little bit of spike or receptor binding domain in the vaccine, it triggers development of these antibodies. And these are neutralizing antibodies. So what they do is they're coating spike, and they're preventing spike from attaching to your host cell and initiating the cascade of infection. Next slide. Okay, a little bit of a primer on how vaccines are developed. So I don't know if everyone on this call is a physician, or a healthcare provider, but essentially vaccine trials happen in 3 phases: phase 1, phase 2, and phase 3. Next slide. So phase 1 is what we saw happen in March. So it was the 20 to 100 healthy volunteers, essentially basically giving them a dose of the vaccine and then doing dose ranging. So giving 1 dose and then giving a higher dose and then trying out the different adjuvants that you would attach to it to see what was the best dose in terms of optimal generation of a protective immune response, neutralizing antibody, and then also the least likely to cause really bad side effects. So sort of hitting that sweet spot between immunogenicity and reactogenicity, which is all of the sort of terrible side effects you get with dose 2. Phase 2 is the next phase of vaccines. It's usually several hundred volunteers. And then this is safety and efficacy. And it's really sort of helping you to understand whether or not these vaccines are safe in larger groups. And phase 3, next slide, is really the one that we think of as sort of the final gold standard. These are the randomized clinical trials where half the people get a vaccine and half the people get a placebo, and then you follow them and then you give them the vaccine, and you're really looking for differences in the number of cases of coronavirus in these 2 groups. And so this is the phase 3 trials for many vaccines, for the mRNAs, for the Astrazeneca, the Janssen, the Novavax, all of those are either in process or completed. And so the decisions about approval for vaccines are really made based on the results of the phase 3 trials. But the important thing to understand is that you don't get to phase 3 unless you've already been through phase 1 and phase 2. So that these are very safe, vetted processes that get you from phase 1 to phase 2 to phase 3 to FDA approval. Okay, next slide. Sorry. This is also a slide courtesy of Barney at NIH. And this is really sort of a close-up just to emphasize that this vaccine was developed very quickly. And that what happened was that there was a first outbreak back in December, on December 31st. And then the sequence of the virus was really posted just a couple of days later from a lab in China. And based on looking at that sequence, they were able to design the vaccine focused on spike. Spike is the portion of the virus that attaches to the host cell. And then they took that spike piece and they basically used it to be a piece of mRNA. They were able to then very quickly move forward and go through animal trials and then get to phase 1 by March 16th. And so that's really lightning speed. The 3 days followed by the 38 days, followed by the 24 days. Next slide. And really, if you compare this to the previous fastest timeline that we've ever had for development of vaccine, this was Zika, and that was 190 days. That's also lightning speed. For RSV, for example, which is the virus I work on most of the time, this vaccine development process has taken 50, 60 years and we're still not at a vaccine. So the fact that this happened this quickly is remarkable. But I think the point to emphasize when you're talking to your patients about how quickly this vaccine is developed and their concerns about safety around the vaccine is that the vaccine development that happened followed all of the correct steps. It followed the preclinical, followed by the phase 1, followed by the phase 2, followed by the phase 3. And that it really sort of occurred in a background of decades and decades of work that had been done already to better understand how coronaviruses infect humans. So it is fast, but it was done safely. Next slide. Okay, I'm going to focus on 3 different types of vaccines. These are the ones that you'll probably hear the most about from your patients and get asked the most about. So I wanted to make sure that we just went over the data on this. So there's the genetic vaccines, which are mRNA vaccines. The ones that we think of are Moderna and Pfizer. There are the viral vectored vaccines. These are adenovirus. So it's a replication incompetent adenovirus. So basically an adenovirus that does not replicate in humans. And it's that adenovirus backbone that the spike protein gene is inserted into. And so you have spike along with an adenovirus. And the advantage of the adenovirus vector is that it grows up very quickly. So you can make a lot of it and it's very cheap to make. And so the adenovirus viral vectored vaccines are the ones that you think of as the Janssen and then the Chimpab, which I think is the AstraZeneca. And the protein subunit vaccines are really what we sort of think of for normal vaccines, like the ones that we give for almost all childhood diseases are protein subunit vaccines. And those are the ones that are coming a little bit later. I think Novavax is a protein subunit vaccine. So mRNA, adenovirus vector, and protein subunit. Those are the 3 that are in play right now. Next slide. Okay, how do mRNA vaccines work? This is a nice schematic from a paper in the New England Journal by Fernando Pollock, which goes over how the Pfizer and the Moderna vaccines work. So essentially here at the top left, you can see that there is a piece of messenger RNA, which is a bit of genetic material, and that it's coated by a lipid nanoparticle. So basically a coat of pieces of fat that are surrounding this genetic piece of material. And what you're doing is you're injecting that whole thing into your arm, and then it goes into it gets taken up, and then it's into the host cell, and then what it does is it expresses pieces that mRNA, which is expressing spike, it makes copies of it, and those copies of it then shoot off onto the surface, and then they get passed on. And so it's basically the spike alone that's being sort of replicated within your body and then causing your body to mount a good, strong immune response. The emergency use authorization for the mRNA vaccines happened in December of 2020. Next slide. Okay, so the dosing of these mRNA vaccines, the Pfizer and the Moderna, they're both 2-dose vaccines. The Pfizer is, I believe, 20 days apart, or no, 21 days apart, and then the Moderna is the 20, I can't remember which one is which, but they all occur 3 to 4 weeks after the first dose. It really doesn't matter. What you're doing is you're trying to prime your immune system. So you're showing it to your immune system the first time. And then your second time is sort of where you're really sort of getting your strong immune response to vaccination. So 3 to 4 weeks after your first dose, you get your second dose. So 2-dose vaccines, both of them are mRNA. They are essentially the exact same pre-fusion spike construct that's in both the Pfizer and the Moderna. They were both originally developed at NIH. Pfizer then moved their platform forward with a different lipid coat, and then Moderna moved theirs forward with a separate lipid coat, but otherwise they're exactly the same thing. Next slide. Because they are exactly the same, they actually had the exact same results. So this is a curve that shows the difference in the number of cases of COVID-19 in individuals who got the vaccine versus got the placebo. So days since randomization is on the x-axis, and the number of cases, or the cumulative incidence, so the overall number of cases over time, are shown on the y-axis. And mRNA is in orange and placebo is in purple. And this is what you want to see. You want to see a nice separation of these two curves. And what you can see here, really interestingly, is that you can see that the curves actually separate out early. So those two arrows down at the bottom left, those are dose 1 and dose 2. And you can see that the separation happens just after dose 1, that you're starting to see a protective effect after just 1 dose, which is certainly amplified after receiving dose 2, but 1 dose is providing some element of protection against coronavirus infection. Next slide. Okay, and then one of the things that we talk a lot about in vaccine trials is, what are you measuring? What is the outcome of importance? Are you preventing infection, which is just coronavirus, PCR positivity, molecular test positive for coronavirus, plus symptoms, or are you trying to prevent severe disease? So severe disease means that you are, sick enough to go in the hospital, you require oxygen, sometimes you're intubated, but sick, really, really sick. And so what we found in these trials, in the Moderna and the Pfizer trial, is that it was protective against both. It was protective against symptomatic disease, and it was particularly incredibly well protected. People were incredibly well protected against severe disease. In fact, across all of the trials, you see that it almost has 100% protection against severe disease. When we were discussing how to power these trials initially, when we were starting the Moderna trial, we had to think about, what is the right thing to do? Is it to power it based, are you trying to enroll enough people to be able to look for outcomes, including death and severe disease? Or are you just trying to enroll enough people so that you can have an outcome of symptomatic disease? Obviously, you need to enroll less people if you have a more common outcome. And if you need to enroll more people if you have a rare outcome. So the decision was made across all of the trials, Pfizer, Moderna, Janssen, Astrazeneca, to be doing all the trials to prevent symptomatic disease. But when you do an analysis of the results of these trials, what you're finding is that not only does it protect against symptomatic disease with an efficacy of 94 to 95%, you are extremely, extremely good at protecting against severe disease with an efficacy of close to 100%. Next slide. Okay. One of the things that we get a lot of questions about is about adverse event profiles. So this is on the top is Moderna, on the bottom is Pfizer. These are both from their New England Journal results. They unfortunately did not ever present the results in a way that you could do a direct comparison. So on the top, I'll just orient you on what this is. So on the top is the injection site adverse events after dose 1 for the Moderna study. So what you can see here, for example, is that 84% of individuals who received the first dose had an adverse reaction at the injection site. So they had a sore arm or a rash or swelling, and as opposed to 20% in the placebo arm. And then after the second dose of the Moderna vaccine, the second panel on the top, you can see that the adverse events, this is now measuring systemic adverse events. So fever, chills, muscle aches, just sort of feeling like you have the flu. That was 80% in the vaccine arm and 37% in the placebo arm. When you look at the bottom at the Pfizer, you're seeing very, very similar numbers. So the first panel on the bottom left is events after the first dose, local events. And the panel on the bottom on the right are events after systemic events after the second dose. What I can tell you is that people often ask, should I get Pfizer or Moderna? Is one going to give me less adverse events than the other one? And the answer is no. It's not. They're almost exactly the same vaccine. The only thing that's different about them is their lipid coat, because one of them is proprietary to Moderna, one of them is proprietary to Pfizer. But essentially, it is the same product being injected into you, and then the second dose is approximately the same amount of time after the first dose. So no, you're going to get the same adverse event profile after either Pfizer or Moderna. So you should not make decisions based on that. The other thing to note about the adverse events is that they tend to go away after 48 hours and that they really don't persist. And there's been actually a lot of follow-up now to support that. Next slide. Okay. I wanted to now talk about the AstraZeneca vaccine, which is probably the next, well, Janssen may go first, but it will be one of the next ones to be approved to receive FDA emergency use authorization. So this is the chimpanzee adenovirus. So it's an adenovirus vector vaccine. So again, so this adenovirus comes from chimpanzees. So it's a non-human adenovirus. It's modified so that it does not replicate. And then what they do is they insert the spike protein into the adenovirus, and then they use it as the vaccine. And essentially, it's the same. Everybody's vaccine targets spike so far. So everybody is making vaccine against spike. It's just a matter of what is holding the spike. So for Moderna and Pfizer, it's naked mRNA, and for the chimp ad Oxford vaccine, it's a chimpanzee adenovirus that's basically replication incompetent, so it won't replicate in humans. Next slide. Okay, so this may come up for you. It's a little bit complicated to explain to your patients, but the main question that often comes up is like, why is AstraZeneca not as good as Moderna and Pfizer? Because if you look at the numbers, here is again sort of your your efficacy analysis of the same curves where people who got the chimpanzee adenovirus are on the bottom, and then the ones who got placebo are on the top, and then you can see a nice separation of the curves, good separation early on. Unfortunately, in this trial, what they found was that there was a clear difference between those who accidentally got a different dose. So those who got a different dose, a low dose instead of the standard dose, followed by the standard dose, had 90% vaccine efficacy. Whereas those who got the vaccine the way it was supposed to be administered, standard dose followed by standard dose, had a 62% vaccine efficacy. A paper just came out this week to try to tease out why that is. Is it because the lower dose was somehow better, which wouldn't make any sense? Or was there something different about these two groups that was not related to the amount of antigen in the first dose? And it turns out that it's the difference in efficacy may be due to the time difference, that actually a longer time between your first and second dose seems to make for a more efficacious vaccine. And so that is still being teased apart, and that's probably why AstraZeneca is not going to receive emergency use authorization that early, because FDA is probably going to require more data before they're going to approve this one because of all the issues with standard dose, low dose, and the differences in efficacy, which are really sort of puzzling for everybody to interpret. Next slide. Okay, so when you compare across the different vaccine candidates and you're looking at efficacy, so you have the mRNAs, the Moderna and the Pfizer, and those are 95%, they're great. You also have Sputnik, which is I think also a spike vaccine from Russia, and that one is also 92%. Sinovac is not great. It's about 50 to 60%. And then this chimp ad, this Oxford AstraZeneca vaccine comes out to 70% if you combine everything together. But as I told you, it's kind of unclear what's driving the difference between the two things. So they're all really good, I think, is the first thing. And then Janssen actually just came out on Monday, and theirs is, I believe, around 60% after dose one, although it's only by press release. They haven't published their paper yet, which is why I didn't want to include it. So these are sort of the efficacy of the different candidates. And then the other thing that drives sort of distribution of these vaccines is cost. And what you can see is that the mRNA vaccines are really much more expensive than production of the other vaccines, the AstraZeneca, and then the Johnson & Johnson, and then the Sputnik vaccine. But it costs much less to produce those vaccines. And a lot of that has to do with the fact that the mRNA technology is much newer. And I think it also has to do with sort of the storage conditions. Next slide. So the big issue with these vaccines is how they need to be stored. So the AstraZeneca one, reason why everybody wants this to get approved as soon as possible is because it's stored at normal fridge temperature, as opposed to Pfizer, which needs to be at -70, and Moderna, which needs to be at -20. And that really sort of, when you think about scale of distribution to places outside of academic medical centers and big cities, that becomes a major issue. So that as we think about, global rollout of these vaccines, we're really not looking at Pfizer and Moderna as our candidates to go globally, that we're really looking at AstraZeneca and Janssen as the global vaccine candidates, because in places outside of the US and other resource-rich countries, to be able to get these vaccines to those places requires a cold chain that's not possible for Pfizer and Moderna. Next slide. Okay, a little bit about sort of reporting of adverse events, and the specific question that came up about allergies and whether or not people with pre-existing allergies should be getting vaccine or previous reactions. So the first thing I want to point out is that there's VSafe, which is a way to register after you get the vaccine, and it captures information about your sort of side effects after vaccination. So I would encourage you to tell your patients to go ahead and do that. As of January 2021, CDC published a report. First, almost 2 million vaccines given in the United States, that there were only 21 cases of anaphylaxis, and that 17 of them had prior allergic reactions to drugs and stings, and then of these, 7 had a prior history of anaphylaxis. So you're not someone who's had a history of allergic reactions or anaphylaxis specifically, you are very, very unlikely. And even if you have, you are still very, very, very unlikely. The rate is 11.1 per million doses. The one thing that the CDC cautions is that those with an allergy to polysorbate or PEG is what makes up that lipid particle around the mRNA vaccines. They're the ones who should not get the mRNA vaccine, because the thought is that it's actually the lipid coat that's causing the anaphylaxis, not the mRNA, because people don't react to naked DNA or RNA. And then again, register after the vaccine at VSafe. Next slide. Okay. One of the questions that comes up a lot as well is about pregnant women and children, and when. So first of all, is it safe for pregnant women to get vaccines? And then when are we going to have the results in the children? So this is actually my area of research, and before COVID, and so we know that pregnant women are very high risk for severe disease to many respiratory viruses. If you remember, 2009 H1N1 was terrible. Was terrible for pregnant women. And then also we think that pregnant women are more likely to get sicker from coronavirus disease. I can tell you that we have a couple of things that we can look back on from previous studies and previous work that has been done with the different constructs. I think some pregnant women are a little bit hesitant to get vaccines because of the fact that they're genetic vaccines, or they just haven't been tested in pregnant women, and that is all true. What I can tell you is that if your pregnant patient has the luxury of being able to decide which vaccine she's going to get, I would say she should get the first one that's available. And if that's not possible to get the Pfizer or the Moderna, and that there are some others that come up, both the Astrazeneca platform and the Novavax platform have been tested in pregnant women for other vaccines. So Astrazeneca platform has been used for Ebola. And then Novavax has a very, very strong record with RSV in pregnant women. They're actually the first company in the world to do RSV vaccine trials in pregnant women. I was actually an investigator on those trials. So they know what they're doing, and they've injected tens of thousands of women with this vaccine, and same with the adenovirus vectored vaccines with AstraZeneca for Ebola. So pregnant women have received the vector, but they haven't received the spike construct. But I don't think the spike construct is going to be the thing that causes an issue. Okay, for children, the question about children is, what do you do with children? And so I think there are a lot of bigger questions around that. I'm happy to take questions about that, but essentially I personally don't believe children are at very high risk for severe disease. They carry virus, they don't tend to be very symptomatic. They can certainly transmit, but they don't transmit with the same efficiency as adults do. So when you're thinking about choosing to make decisions based on whether or not your child is already vaccinated, I personally would not wait for that, because I don't think most children are going to get very sick from coronavirus, even if they get it. And they seem to be getting it at very low rates. And when they get it, their hospitalization rates are extraordinarily low. There is multi-system inflammatory syndrome, which I hope somebody else has time to talk about, but it's a very, very, very rare event. So the trials that have started in pregnant women and in children, so let me see. So Janssen and Pfizer and Moderna, I think, are all starting pregnant women trials. I'm helping out with some of those. And then Seattle Children's is actually running a lot of the pediatric trials. They'll first go down to 12 to 18, and then they'll go below 12. Really, the goal of these trials is establish safety and immunogenicity, so like phase 2 level trials, and they will not probably go to phase 3 based on what we're talking about at NIH right now, but that may change. That most likely what we'll do is rely on the phase 2 data to make the decisions about whether or not to allow these to be used in children. Okay, next slide. Okay. Okay, so the other question that came up, I think, before the talk was about reduction in overall transmission in the community and achievement of herd immunity. And that's also a really big question. So I would just say that in general, so herd immunity is basically the proportion of the population you need to achieve protection in either through vaccination or natural infection to be able to protect the unvaccinated in the population. And that this herd immunity is actually a number. It's 1 over 1 minus R naught. So it's inversely proportional to your R naught. So the more transmissible a pathogen, the harder it is to achieve herd immunity. So as the R naught increases, so R naught for measles, for example, runs between 10 to 15, you need a higher coverage of your community to be able to achieve that R naught. But the lower your R naught, the less you need to vaccinate. For coronavirus, this seems to be an ever-moving target. It's probably somewhere between 60 and 80, but the dynamics have just changed with the new variants that may drive up the herd immunity threshold a little bit higher. Okay. So, and then the question about whether or not getting a vaccine protects you from asymptomatic infection and then further transmission. So this is what we know. We know vaccines prevent symptomatic disease. I would say that vaccines, and this is just my opinion, are likely protective against asymptomatic infection. So those who carry the virus but are not sick, but it is not going to be as effective as for symptomatic infection, because really to protect against asymptomatic infection, you need to achieve something called sterilizing immunity, which basically means your antibody titers have to be so high that you can't even acquire the virus in your nose. And that, I think, is a very, very high bar, and most vaccines do not achieve that bar. So, however, the fact that it is so good at protecting against symptomatic infection, and it's probably going to be pretty good at protecting against asymptomatic infection, means that once we achieve a certain proportion of the population that's vaccinated, we're going to start seeing interruption of transmission. And we're already seeing this in Israel, in the nursing homes. Deaths are going down. And the reason why deaths are going down is because you have protection of the vulnerable, and then you're also probably reducing some element of transmission. Okay, next slide. Here I've compiled for you all of the data that is published to date from the different trials about how effective this vaccine is, the vaccine efficacy against asymptomatic infection. So the background first is that, you know, it's 95% effective against symptomatic infection, but above and beyond that, does it also protect against asymptomatic? So for Moderna vaccine, after 1 dose, they did sampling of people before they got their second dose, because they wanted to know how many people going into their second dose had acquired coronavirus infection. And that they found that 39 people in the placebo arm had evidence of asymptomatic infection after the first dose, whereas 15 people in the vaccine arm had evidence of asymptomatic infection after the first dose, which led to a vaccine efficacy of 62%. AstraZeneca's, you know, it's kind of a mess because of their low dose, high dose issue. But essentially what you can see here is that overall from the 2 trials that have published their data, that you are seeing some element of protection against asymptomatic infection. But that we don't know what's going on with Pfizer, with Novavax, with Sputnik, or with Johnson & Johnson. But usually the way that this comes up is that in their FDA filing, they have to show this data, usually in one of their supplemental tables, and Johnson & Johnson is about to file. So I think we'll know this, at least for Johnson & Johnson, pretty soon. Pfizer did not. I went through their 100-page document, and I couldn't find any evidence of their looking for this as part of their study. And then I talked to the PI of the Pfizer trial, and he said that they didn't do this, but that they're doing it now. And so we should have some data, but it's not going to be available for a while. Next slide. Okay, the emergence of new variants. So I know that they've been renamed a couple of times. It's a little bit confusing. So the new variants of concern are these new variants of coronavirus that are spreading throughout the UK, through Brazil and South Africa. And now we found at least N501Y, which is the UK variant. We found it here in Seattle. So there's some evidence that these variants are more transmissible, but it is still unclear whether or not they cause more severe disease. And here on the graph, and this is now, of course, outdated because it was 2 weeks ago, you can see that the number of cases really spiked in Ireland and Britain, and that was because of the emergence of the 501Y variant. Next slide. Okay, so these are data, very, very, very early data from the Moderna trial. And this might be a little bit too much, so I'll just ask me questions if it gets confusing. But basically, on the bottom is the different viruses with the variants. Okay, so 501Y here is the UK variant, the one that's now here in Seattle. This 417N44K and 501Y is a virus that has acquired some other mutations to spike. And then the B1351 is the South Africa variant, the one that we're very worried about. So what you can see here on the y-axis is antibody titer. Basically, how well does antibody neutralize these new virus variants? And here, the higher the number, the better you do. So basically, for wild type virus without mutations, you can see that that you neutralize pretty well, that your body is able to control. But as you go along and this virus acquires more and more variants, your body is not doing so well anymore. These are the results after vaccine, the Mrna vaccine. We also have results that we've now probed against natural infection here in Seattle, and that was just published in Cell yesterday. And that we also see the same effect that natural infection or vaccination, it's becoming obvious that those are not fully protective against the South Africa B1351 variant. That's not good, and that's why we're worried. But I think what it means is that we need to do a really, really good job of basically controlling the infection and controlling transmission, because viruses need to transmit to mutate. So the higher coverage with vaccination that we can achieve, and the better we do at stopping person-to-person transmission, the less likely we're going to see the spread of these variants. And then the thing that emerged very recently, next slide, is that actually, I think this came out yesterday, that they looked at Novavax, which happened to be a trial that was running while the new variants were emerging. And unfortunately for Novavax, they were running their trial in South Africa and then the UK. And what I think this is actually really good. It shows that even in South Africa, where this variant is the predominant variant that's circulating in the population, that vaccine efficacy was still 60% when you excluded the HIV infecteds. So it's not zero, which is good. And 60% was actually the bar that FDA wanted us to achieve when we did these vaccine trials. And that in the UK, that with that 501Y variant, the vaccine efficacy was 89%. So I think the take-home message here is that it's good news that vaccines still have a VE of 60 to 89% against the new variants, but that these vaccines will likely need to be updated over time and booster shots will need to be given. And potentially we will be heading towards an annual vaccine like for flu. The good thing about mRNA or genetic vaccines is that genetic vaccines tend to be, you can change them very quickly. And they're all spiked. So basically you just plop in 44K and you put that into your next vaccine dose, and then you will achieve protection. It's going to always be a race against the virus though. And that's the worry, is that we are going to, it's an arms race essentially. And so what we need to do is basically achieve enough herd immunity by enough vaccination that we don't have to engage in this arms race, that we can get to a point where the virus is not spreading fast enough in an uncontrolled way that it has the ability to acquire these variants and for them to then take over a population. Next slide. Okay, so I wanted to just make a point that there are, that vaccines are getting rolled out, but that there's unequal rollout, that many countries are not getting vaccines. This is due to a variety of reasons. A lot of it has to do right now with cost, and then also with the fact that some vaccines, the ones that are approved, cannot be used in these places because there's no cold chain. Next slide. Some sources of reliable vaccine information are here. The CDC website and the Washington State website, I think, are both really, really good if you want to point your patients to those. Next slide. And then some conclusions and future directions. So I think that, first of all, these vaccines were developed in record time, and that will come up as a concern from your patients. But the point to emphasize is that all of the appropriate steps were taken, and that it really relies on the backbone of science that's been done over the last 2 decades. The mRNA vaccines are efficacious, 95%. Many of the other ones, Johnson & Johnson and AstraZeneca, are really good. There's a difference in cost and storage conditions across the candidates. We don't know the full effect on reduction of asymptomatic transmission, but these studies are coming. We will likely need to update for new variants. The things that are still missing that are being done now, so long-term safety data, FDA requires 2 years, so that those long-term studies are being conducted. The data in pregnant women and children are still yet to come. And then this question about 1 dose or split dose, that's also under a lot of debate. We actually tried to do that study, but then it did not start, so we're not doing that study anymore. But I think you know, hopefully that study will get done, maybe not by NIH, but by a different group, to understand whether or not a 1 dose strategy will be okay. Okay, next slide. And that's it. So feel free to email me with any questions. This slide deck is available for anyone who needs it, and I'm happy to answer questions.
Dr. Chu, thank you so much for your presentation. This is Mrna Vaccines. I think what we'll do is we'll take questions in the chat box, and then we'll be able to answer them towards the end. I know that these slides are available to the public or to our WAPI and Indian American Community Services members as well. So hopefully they'll be able to reach out if they have further questions, but I'm also hoping we can answer some of their questions in the panel discussion at the end, just to save on time this morning.
Okay, thank you.
We're going to move on, and I just wanted to welcome Dr. Rayna Patel, who is an assistant professor in the Division of Allergy and Infectious Diseases and the Department of Medicine at the University of Washington. And give us 1 second as I just allow her to take over screen sharing.
Thank you, Amish. Can you hear me okay?
Yes, we can.
Great. Let's see, can I advance the slides? Yes, it looks like I'll be able to. Great, perfect. Thank you to WAPI and Indian American Community Services for inviting me to talk today. It is honestly my distinct privilege to be able to talk to you as an Indian American who is so excited to be giving back in a small way to our South Asian American community. And again, a huge thanks to Helen Shu, who's been a true warrior in this work around COVID. It's been a pleasure to me working at the same institution to, you know, when early on we got so many questions, I was able to say, we actually have a coronavirus expert at our institution who's been helping us tremendously. So thank you, Helen, for all the work you do. Looks like okay, there. So today I am going to talk about what we know and what we don't know, and it's all really incredible what we do. But I'm going to focus on some basic terminology, background, and go into epidemiology, and then focus on special populations that are affected. Amish, it does look like there's a major lag in my control. So yeah, perfect. If you advance, I would greatly appreciate it. So I just wanted to put a really big picture here with all the different plagues, epidemics, and pandemics, and pandemic implies a global nature to an epidemic, that started from as far as human record goes. And as you can see, the orange, the size of the orange dot indicates the density. And so the largest pandemic that we've experienced is the Spanish flu that occurred at the turn of the 20th century, with over with a death estimated over 100 to approximately 100 million. And as of September, for COVID-19, we had reached about 1 million. And as you all know, we have surpassed that number now. So we're getting close to being a very large pandemic. Next slide. Here's a visual timeline of the pandemic. As Helen mentioned, this likely was circulating in China as early as November, some documentation as early as November, but officially was reported to the WHO on December 31st. And then within the first few days, the genetic coding of the virus was conducted, and that's what's really speeded the vaccine development. And then within the US, and we here in Washington State, we're in the unfortunate but privileged position to be reporting the first US case in a returning traveler from China. And as well, by the end of February, had reported the first death in the US. And after that point, you've seen that this pandemic just blossomed worldwide. Every part of the world has been affected. Next slide. I wanted to show this snapshot from the Johns Hopkins COVID dashboard that many of you may have accessed during the pandemic. And I left it here. This is the shot I took on March 17, 2020. So at that time, here in the top corner, you can see that there were less than 200,000 total cases with only about 8,000 deaths reported. Next slide. And then when I took a snapshot of the same dashboard last week in preparation for this talk, we have over 100 million cases reported with over 2 million deaths. This is a really profound evolution of this pandemic. Next slide. And what I want to show to you here are the different curves for different countries. And so it's been very different how the pandemic has affected various regions and countries. And the green line here are the surges for the US. And so there are 3 distinct surges when it first, when we first detected in March, April, nearly a year ago, then over our summer, and then the 3rd surge that started in our late fall. And India, which is this light blue line, has really only had 1 surge, so to speak of so far. India started detecting its first cases as well starting in March and April, but didn't really peak until September. And when it peaked in September, the entire global community was extremely worried. Knowing the population density in India and its already fragile healthcare system, there was a lot of concern that this epidemic there would just blow out of proportions and decimate any progress made India in the last 3 to 4 decades. And India, remarkably, has managed to get its epidemic curve down to the point where it's much lower than many of the other countries. And the other country that's been most affected by COVID-19 is Brazil, which is in this orange line. And in Brazil, it's a very interesting local picture where there are certain parts of Brazil that are dramatically affected repeatedly over time. Next slide. And so these are the cumulative cases by date. And again, as you can appreciate, in the green light is the curve for the US, which just keeps getting larger and larger. In moments, the steepness changes. But in India, that curve has been relatively low. Next slide. And this is to look at the impact or the burden by death. And so here in different colors, you can see, and the darker green is North America, where US most definitely has a disproportionate burden. Ironically, as our former President Trump used to say, America is number 1 first in this. And then Brazil has the next largest burden of death, even when adjusted for population size. But India has very little, and some of this is, most of this in Asia is outside of India. Next slide. And so this has begged a question that I imagine many of you have been thinking about and and discussing with your friends and family and colleagues who are back in India. Why and how is India doing so well in controlling COVID-19? And some of you have may read this article that was just published this past week in NPR. And it's not, to be fundamentally honest, it's not clear why. There are some things that are working really well in India, such as mask mandates. So India is one of the countries in the world that has done a national level mandate that has been followed up with really concrete reinforcement. You know, my father, who has been living in Khambhat, Gujarat through this entire pandemic, you know, tells me every now and then, oh yeah, there are police out at the, you know, road intersections that are literally pulling people over and fining them. There are also other interesting social distancing type mandates in India. For example, some places in India earlier on in the epidemic required individuals who were positive to go into COVID-specific quarantine centers. Other places encourage individuals to go there. And I can't tell you how many of my family members access those resources. And most recently, India has implemented a strict quarantine after travel, after international travel. So when coming back from an international flight, you are required to enter one of the quarantine approved locations to stay for the next 14 days. Some of those details are shifting, the number of days, et cetera, but these are really profound, nationally coordinated mandates that India has been able to implement that we here in the US have not to date. Many people have credited India for having consistent messaging about ways to fight this pandemic. For example, Narendra Modi has been early on endorsed mask wearing, and when he appears in public, consistently wears masks and so forth. There are some biologic possibilities here. We don't know the answers to questions like, are the variants of SARS-CoV-2, the virus that are circulating in India, more or less virulent compared to other places? Could the hotter climate or more humid climate make a difference? There are these hypotheses that more humid air weighs down the particles of the virus and makes them less transmissible. So some people argue that may be playing a role, and why in general we haven't seen this pandemic blow out of proportions in many tropical parts of the earth. But others argue that there is so much more air pollution, which you know, not only predisposes people's lungs to insults that may make them more susceptible to a COVID-19 infection, but also that may play a role in how the viral particles are physically and physiologically transmitted. People argue, okay, well, if it's really hot outside, it forces more people indoors, so they may be congregating more. So it's not quite clear what exactly may be happening there. People have indicated that India has a larger, younger population than some of the other westernized countries like US and those countries in Europe. Could that be making a difference? Possibly, but even age adjusted estimates are lower in India, not just for test positivity, but also for deaths. Could there be, people have indicated a survivorship bias, you know, basically to be able to survive in a tough climate. In India, you know, those people who are living now have likely survived all the odds and therefore have more robust, you know, all the way from biologic systems, immune systems to socioeconomic benefits. And there's this really leading paradigm that I can't tell you how many times I've been discussing with my cousins in India who wonder if their exposure to more pathogens over time has given them more robust immune systems, and that helps protect them against COVID. I personally am highly skeptical of that proposition because I'm not an immunologist, but my belief is that viral immunology is very different than parasitic immunology, for example, from malaria or even gastrointestinal parasites. So I'm highly skeptical of that. And then what's one proposition that I am a little more interested is in this idea of herd immunity. And so a seropositive study in Delhi conducted many months ago showed that nearly 50% of the individuals had antibodies positive for COVID-19. And we know that clearly case positivity rates are much lower than that in Delhi. So maybe there have been enough people, healthy people who've been infected with COVID that in a hidden sense, communities in India, because of the population density and so forth, are starting to develop herd immunity. Next slide. But I really welcome all your thoughts and questions on this because there are no concrete data guiding us. I wanted to piggyback on Helen's points about new variants here in terms of epidemiology as well. So here is a snapshot from the Institute of Health Metrics and Evaluation, IHME modeling. This is best done with the real-time use of their website, but I just took a snapshot to show you here's the red line for India and the blue line for the US. And when accounting for potential increased transmissibility of the new variants, in India, we don't really see a huge impact of that. So the line remains pretty slow. In the US, that line, we still predict that there will be a lower incidence of new cases in the US, but this line is a little higher with the new variant than it would be with the old variant, which would be lower. Next slide. So I want to take just a few moments to talk about some special populations and highlight inequities in that work. Next slide. So those of us who are living in the US hear this probably every day, where people of color are disproportionately affected by the pandemic. So these are test positivity rates. And as you can see, it's a representation of the density per 100-person people living. So then the pandemic is disproportionately affecting Black or African Americans, Indian or Native Americans, and Latino persons compared to everyone else. Next slide. And this data is borne out by our local information. So this is from our King County, Washington dashboard that I downloaded last week. And here you can see in the blue line are the percent of cases, and in the gray line are the percent of the county population. And so for the white residents, the test positivity is much lower. But when you look at Black, look at that, that's nearly 2 times higher among the Black populations, as well as 2 to 2.5 times higher in the Latino population. Next slide. Another very extremely interesting phenomena is that men or males appear to be more likely to be affected by COVID than women or females. And this is really, again, counterintuitive because for most aspects of diseases or infections, women or females are more disproportionately affected. And here you can see are the data. This is very early on in the epidemic, but this trend has continued. What I will note is it's very interesting. India is one of the only few countries, it's I think only one of 3 countries so far that has bucked this trend, where the death rate is slightly higher in females than males. The total death rate is very low in India, but there is a slight flipping of that data. But just about every country in the world is reporting higher death, severity of COVID infection, and in some cases, even incidence of COVID to be higher in males and females. Next slide. And what I want to do here is just show some primary data from a project I'm involved with a PhD student here at University of Washington. So we looked at the association of sex with COVID-19 outcomes who are receiving SARS-CoV-2 testing at the University of Washington Medical System through August of last year. And here you can see in the very last column, that the incident rate ratio is higher for males than females for testing positive, for getting hospitalized, for having severe COVID, and for dying from COVID-19 related death. Next slide. My colleague Helen Shu mentioned pregnancy, and I wanted to show this data from our local area. This is COVID-19 pregnancy registry in Washington State, and here they've shown and it's retrospective and passive reporting, so many limitations, but they're showing that hospitalization rates for women who are pregnant with COVID versus age-matched non-pregnant women and men is 3 times higher for hospitalization and up to 13 times higher for death. So this early on in the pandemic, there were a few case reports from China and people were like, oh, pregnant women don't appear to be disproportionately affected by COVID-19. That is definitely not the case in the US. Our pregnant women are at increased risk for infection, hospitalization, and more severe outcomes with COVID-19. Next slide. I am a HIV provider, so I wanted to present this slide here. For people living with HIV, it's as if 2 epidemics are coming together and even further confounding or compounding the inequities they face in healthcare. So here's work that a colleague of mine and we're doing in a national data set, where in blue are those people who are living with COVID living with HIV who tested positive for COVID. Then in the lighter gray are those individuals who are not living with HIV and tested negative. And then in the darker gray are those people, again, living with HIV, but not COVID positive. So you can see for non-Hispanic whites, the COVID pandemic is affecting them less. And when you look at Blacks and Latinos, even though they are more disproportionately affected by HIV epidemic, they're even further affected by the COVID pandemic. Next slide. So I'm going to stop here and just end with this a big picture that ironically, we have more questions and answers than we did a year ago. But what is really phenomenal is we are able to ask these questions as a larger community, both ranging from basic science to translational to clinical research to public health and policy. It's just been so phenomenal to see the global world come together and collaborations blossom. So on the top are what I am calling, what I think is the fastest knowledge generation effort ever in history. We are seeing hundreds of publications a day. And as Helen mentioned, the trials, not just for vaccine, but for treatment or prevention are occurring at rapid speed. And a lot of this is not because we're cutting the corners on the science, not at all. It's because there's so much collaboration and coordination taking place all the way from the scientific to the regulatory to, donors, et cetera. And I think one thing that we can say more unequivocally is that we know how to break the transmission chain at the interpersonal level. For example, efforts such as wearing face masks, physical or social distancing, et cetera, are really effective strategies. Things we still don't know. So bigger picture, we still don't quite have our finger on the pulse in terms of transmission dynamics. There is still a question of how much of the epidemic burden is because of asymptomatic infections or carriers, what role children play. It's likely a smaller role than we previously anticipated, and that's why there is this large effort to start bringing children back to in-person schools. We do have greater information that special populations such as women or people living with immunocompromised states are at greater risk of infection or severity of disease, but we still don't understand how or why. And we certainly don't understand how or why some people recover better than others. There's this term called long COVID now, where people are having really long-term sequelae of their COVID infections. And we really are only beginning to even scratch the surface on that. As I alluded to in the epi portion of my talk, how are some countries, states, and counties, even at the county level, recovering better than others, controlling the epidemic better than others? We have some sense of that, but I don't think we know enough. And then all the larger picture, social, political, economic, and moral consequences. I want to highlight the moral part because I know Helen touched on this element in terms of vaccine distribution. The director of the WHO last month said, as a global community, we are at the brink of moral failure. And I can't stress that enough, how that is playing out in COVID vaccinations, as well as in all other elements of COVID, including treatment, diagnostics, and so forth. Next slide. So I will plan to stop there, and I purposefully had tried to keep my portion of it less than 30 minutes. Amish, you can let me know if there's time for questions now, or if you want to save them for later.
I think so that we can stay on schedule, what I'll ask is if people can use the chat box to send any questions they might have specifically for Dr. Patel, but Dr. Patel will also be on our panel after our next talk as well. So you can also, she'll also be able to answer questions out to the audience as well. So if you have a burning question, feel free to send it via text message. I know that she's been responding and we're very grateful for her responding to text messages or chat messages as they appear also. But I want to move on and invite Dr. Malhotra to speak. And Dr. Malhotra, if it's okay with you, I'm going to just help with controlling the slides and moving them as you say next slide.
Yeah. Or Amish, how about, let me just try, I have my file open too, so I can just try and share it here and see if that works.
I think just for the sake of, I think it might be a little bit too complicated to go in and out if you're okay, Dr. Malhotra. So I can just move on with the slides if you're all right.
Okay.
So I want to introduce Dr. Uma Malhotra, who is a clinical associate professor at the University of Washington, as well as with a physician with an infectious diseases specialist within our Division of Infectious Diseases at Virginia Mason Franciscan Health. We're very grateful to you, Dr. Malhotra, also for joining us today.
Okay, thank you, Amish, for the introduction. And I want to thank the organizers for this kind invitation. I'm very glad to be here. Next. So I will be covering diagnostics, the types of diagnostic tests, including virologic and serologic tests, specimen types. Next. Antiviral drugs, immunomodulators, long haulers, and finally, I'm going to touch upon the role for the Ayush Ministry. Next. So I'm going to briefly touch upon the clinical picture just to lay into context the remainder of my talk on diagnostics and therapeutics. As most of you may know, that majority of patients with COVID actually experience only mild illness or asymptomatic illness. And there is a small portion that experience severe disease, and yet a very small portion that experience critical illness requiring intensive care management. And majority of patients experience respiratory symptoms and flu-like symptoms, but it's important to remember that a small portion may experience only GI symptoms such as nausea, vomiting, diarrhea, and others, especially with milder illness, may have just isolated changes in taste and smell. Next. And patients with severe disease exhibit two phases to their illness. So just in the recent week, I admitted 2 patients to the hospital who explained that they were actually feeling much better by the end of their first week of illness, and then started experiencing severe shortness of breath. And so that part of the illness appears to be really driven by the host inflammatory response. And that's when patients present with severe illness to the hospital. Next. And these patients have a variety of manifestations besides their ARDS. They have cardiac dysfunction. They may have myocardial ischemia, arrhythmias. They may have, massive venous and arterial clots. They may present with critical limb ischemia. They may have kidney injury. They may present with strokes. Next. Patients with severe disease, will frequently show lymphopenia. So if in your outpatient setting you see a patient, presenting with lymphopenia, that may be a prognostic indicator that they may be headed towards a more severe illness. So that can be a simple test that gives a hint of upcoming severe illness. And patients with severe disease may have abnormal liver functions and elevated inflammatory markers. Next. So I'm going to now move on to diagnostic tests, and I will be reviewing both virus detection assays and antibody detection assays, which are called serology tests. Next. So the virus detection assays are really the choice for patients presenting with acute illness. And these consist of either a rapid antigen test or a PCR-based test. And as you can tell, that a PCR-based test, is a lot more sensitive than the rapid antigen test. And the antibody tests actually really don't become really peak at about 3 weeks after onset of illness and don't have any role for diagnosing acute infection. Next. And the antigen tests, as the name suggests, are really detecting virus protein. And these are rapid and inexpensive tests, but they have quite limited sensitivity, ranging depending on the type of test, could be as low as 30%, although the specificity remains quite good. Next. And then there are the PCR tests, which are really detecting nucleic acid or the RNA in this case of this virus. And these do require more equipment, typically run in hospital laboratories. They're time intensive, expensive. Next. And there are, at this point, several hundred such assays that are FDA approved, and these assays have high sensitivity and as well as high specificity. Next. And then, as opposed to the virus detection tests, there are the serology tests that are really looking at your immune response to the virus. Next. And there are a variety of these tests, from the simplest type of point of care test, which shows these bands which are positive or negative, and that's a lateral flow assay. And then are little more sophisticated ELISA or binding assays that have somewhat improved accuracy. And then of course are the more complex neutralization assays that show whether the antibodies actually neutralize the virus, but those assays have very limited availability. Next. So this here shows that how some of these antibody or serologic assays can have very low sensitivity, as well as some of them may have low specificity as well. And the low sensitivity could be as to the timing of the test, and specificity issues related to whether the antibody test may actually cross react with other cold coronaviruses that cause cold. Next. So the bottom line I wanted to highlight is that none of the tests that we have, even for the virus detection, is perfect. And there is fair amount likelihood of a false negative test, that is that a person has an infection but the test is unable to detect the infection. And so our tests, unfortunately, cannot say 100%, even if a patient is feeling ill and the test is negative, they cannot say with 100% surety that the person does not have COVID infection. Next. So there may be a role for doing a repeat test if the clinical suspicion is high. And so we'll see, now and then patients presenting to the hospital that have, look like classic COVID and we do a test and the test is negative, then we may actually ask for another test to be run. It could be the same test, or it could be, one of a different test that we may have on our panel to, so to obtain some more diagnostic accuracy. Next. So what are the types of specimens that we use for testing? And you may have heard about the nasopharyngeal swabs, and these are clearly uncomfortable because the swab is going far back into the nasopharynx. Next, please. And over the months since the outbreak, we have a number of other specimens that have been tested and shown to work, and these are oropharyngeal samples. They are nasal mid-turbinate samples, anterior nares samples, as well as saliva samples. And the beauty with some of these other samples is that these can be collected by the patient themselves. Next, please. And the CDC website actually has a nice description of how these samples can be collected. So for example, the nasal swab really needs to go in just about quarter inch up the nares to be collected. So it's rather easy to collect. Next, please. And so I think the bottom line from this section is that the nucleic acid amplification tests really are the tests of choice in the acute setting, although rapid tests, because they're easily available and more available in the community, have a role. And in some cases, it may be that one needs to do a repeat test if there is a negative test and there is a high index of suspicion. So clearly good to go ahead and do a repeat test, because none of these tests are really 100% sensitive. And I wanted to emphasize that we frequently get requests from patients asking for a serologic test to understand if they may have prior infection and if that may have given them immunity. So really it's recommended not to use the serologic tests to either make assessments regarding prior infection or assessment regarding protection against future infections. Next, please. So with this, I'm going to move on to the therapeutics. So as I described earlier, that COVID illness is really a two-phasic illness in patients who present with severe disease. And there is the viral phase, and majority of the patients recover from the viral phase and do not go on to develop any further symptoms. But then there is a portion that develop what we call a hyperinflammatory response. And that is what is causing their severe illness. And these are the patients that present with severe symptoms and require the use of immunomodulators to modify their immune response. And the role of antiviral drugs appears to be really, early on in the illness. Next, please. So here is the SARS-CoV viral life cycle that's occurring, within the cell, like a cell in the lung epithelium. And so a lot of the antiviral drugs will really try to interrupt the virus replication within the cell. And then there are the immunomodulators, which are trying to modulate the immune response. So if there is a hyperinflammatory response going on, these immunomodulators will actually try to dampen that immune response. And among the various antiviral drugs that have been tested, as you will see, remdesivir is the only one that has stood out to be possibly providing some benefit. Next, please. And a number of trials were conducted early on, and the trial by NIAID or NIH, was a double-blind randomized trial that looked at remdesivir versus placebo. Next, please. And this was in patients with hypoxia, that is patients who were requiring oxygen, and it showed that the use of remdesivir in these patients provided improvement in time to recovery and perhaps mortality benefit. Next, please. And these data rapidly led on to issuance of emergency use authorization for remdesivir in the US and ultimately to FDA approval. Next. And this is referred to as a simple study that was sponsored by Gilead, and this showed that majority of the benefit from remdesivir really appeared to be in people who presented within the first 10 days of their illness. And as you will recall, we saw that patients, this early phase is when the virus appears to be what is causing harm. So it wasn't surprising that the antiviral drug is beneficial early in the illness. And it appeared that a short 5-day course was just as effective as a 10-day course that was also tested. Next, please. Just about this time, WHO also released results of their Solidarity multi-country, multi-center trials, and those were actually rather disappointing and showed that actually remdesivir in their studies did not provide any mortality benefit, as also hydroxychloroquine, which was another drug being tested, did not provide any mortality benefit. Next, please. And other drugs that were being tested, such as HIV, drugs used for HIV treatment, lopinavir, ritonavir, as well as some interferons, none of these showed any benefit either. Next, please. And so bottom line with remdesivir is, till date, it remains the only drug that has potential benefit in patients with severe disease. That is patients, who are requiring oxygen, and that is within 10 days of symptom onset, and treatment duration 5 days is as good as 10 days, and it's being used for patients in the US. However, WHO, based on their Solidarity trials, actually does not recommend use of this drug. Next, please. NIH, does endorse use of the drug in patients, hospitalized patients who require oxygen. However, it does not endorse use of any other antiviral drugs, chloroquine, hydroxychloroquine, azithromycin, ivermectin, unless these are used within the context of clinical trials. Next, please. So with this, I'm going to move on to use of immunomodulators. So the real big game changer here was the recovery trial. And this recovery trial tested the use of dexamethasone and was a very this was a very well-designed randomized study that showed and that tested dexamethasone in patients who were stratified based on not requiring any oxygen, requiring oxygen, and those on ventilators. And it was seen that the greatest benefit of the drug was in patients requiring ventilators. So these patients typically have a 40% mortality rate. And this drug, the dexamethasone, reduced the mortality rate to 28%. And that's about a 30% reduction in mortality. Next, please. And this again here shows that the benefit is really in patients who are requiring oxygen. So the point I want to make, and that comes up very often, is people asking or providers asking whether they can use dexamethasone in patients who they are managing in the outpatient basis. And the key here is that actually the medication may be deleterious and really make the survival worse. So we definitely don't want to use this drug in patients who are not hypoxic. And the reason behind that is that these patients who are not hypoxic are actually likely mounting a really good immune response that's controlling their virus, and we don't want to dampen that immune response. So we want to let that immune response work. Next, please. And besides dexamethasone, another group of drugs that received a lot of attention were drugs that have been used in various autoimmune diseases and hematologic conditions. And these are blockers of the interleukin-6 cytokine or its receptor. And this was thought because that hyperinflammatory response, that IL-6 may be a cytokine that's playing a huge part here. But unfortunately, none of these drugs have really shown any definitive benefit in these patients, and it's not recommended that they be used outside of any clinical trials. Next, please. So here, NIH, again, their guidelines really recommends the use of dexamethasone with or without remdesivir in patients with hospitalized disease requiring oxygen, and other immunomodulators are not recommended at this time unless they're in the context of a clinical trial. Next, please. And another therapeutic that received much hype is convalescent plasma that's obtained from patients who have recovered from illness. And this plasma is then given to patients who have acute disease. Next, please. But none of the trials have again shown any definitive benefit, so it is not recommended that the plasma be used. There has been a small trial that showed that plasma with very high levels of neutralizing antibodies may be efficacious in people presenting within the first 3 days of onset of illness. So that's been sort of tricky to identify that population. And so widespread use of the plasma is not being done at this point. Next, please. And another therapeutic that's been in the news lately has been, various antibody combinations that are thought to bind with the spike protein. And so these were initially tested in hospitalized patients, and it was shown that they were actually did not provide any benefit. And recent trials show that the only role for these antibodies may be in people who are not hospitalized, so they're early in the illness, and the role is primarily in patients who may be at risk for severe disease that have a variety of comorbidities like obesity, diabetes, etc. And again, the use of these drugs remains limited because it's not so easy to identify this population. So again, the use remains quite limited. Next, please. So as I mentioned earlier, these patients are really at high risk for blood clots. So we will see, massive DVTs, deep vein thromboses, arterial thromboses. So it's strongly recommended that patients who are admitted receive DVT prophylaxis. A question that I often get from providers is, what about non-hospitalized patients with COVID? Should they get prophylaxis too? So at this point, this is not recommended for non-hospitalized patients. Next, please.
Yeah.
And again, I frequently get calls about, people having an exposure and wondering, whether there is a drug that they can take. And early on, there were trials of hydroxychloroquine and chloroquine, but none of those have panned out. So really, the key is to just, monitor, and monitoring oxygen saturations can be a good idea as well. But there is really no medication that's available at this point to prevent disease. Next, please. So I wanted to highlight that NIH has what is a living document. So it's a really good resource that folks can access and see, what the current recommendations are for various stages of disease. And I'm going to just, again, to bring the summary of treatment all together for patients with mild to moderate disease who are not hospitalized. There is really no medication recommended at this point. People who are at high risk for severe disease, they may be able to access, through their physician, a variety of antibody combos. But other than that, key for them is really monitoring their oxygenation at home. And, as many of you may have heard about, silent hypoxia. So oftentimes patients can get severely ill and not know that they are ill because they don't experience shortness of breath during COVID. So I think that's the best thing that can be done is monitor their oxygen saturations and present to a hospital if their oxygen saturations are low. For patients who are hospitalized, but not requiring any oxygen, there may be a role for remdesivir if they're within 10 days. Next, please. And patients who are hospitalized requiring oxygen, there is again a strong role for dexamethasone plus minus remdesivir, depending on duration of illness. Next, please. So a question that often comes up is duration of quarantine. how long is somebody infectious. And it's important to understand that the virus testing, that the NP swab that we do using PCR, I showed that earlier on, it's a highly sensitive assay, and it may actually remain positive for months. And so it doesn't mean that the individual is infectious. It just means that they have some virus, particles or virus, RNA hanging in their nose, and that's just remaining positive. So now we have really clear guidelines from CDC that the highly infectious phase is really about 10 days from symptom onset. So that's when patients need to be quarantined. It may be in some, rare situations if people are immunocompromised, they have some, underlying malignancy or other immunodeficiencies where we may need to extend that period to 20 days. And then there are the patients who never develop any symptoms, and those patients, they just test positive. They just need to be quarantined for 10 days after their positive test. Next, please. And so the post-COVID syndrome, what is the post-COVID syndrome or the long haulers? This is when patients continue to experience symptoms past the first 3 weeks of onset of illness. And there are about 10% of patients that may experience prolonged illness, and there is, limited evidence as to how these patients must be managed. And most recover spontaneously with holistic support, rest, symptom management, and that's really, I think, what is needed for these patients. And home pulse oximetry may be again helpful because some patients may, continue to experience some breathlessness. But however, we do need to continue to maintain, vigilance for, particularly for cardiac and neurologic complications. I've seen plenty of patients who present, like a month after COVID and present with cardiac ischemia or present with a stroke. And, that can be that they're, in this hyperinflammatory phase and they have, high levels of D dimers, etc. So it's, important to be alert for those complications as well. Next, please. And a good resource that I want to point out to you is in the British Medical uh, Journal. And I think, next, please. Okay, I'm sorry that the reference is not showing here, but I think if you just Google, there is a schema in the if you Google post-COVID syndrome and British Medical Journal. So it really kind of walks you through in terms of workup, but the bottom line still ends up being continued symptom management and lifestyle modifications in terms of rest and good nutrition and really self-pacing. Next, please. And I wanted to spend a couple moments on COVID-19 in India and the role of the Ayush ministry. So Ayush, kind of interesting acronym that stands for really all of the non-allopathic treatments for Ayurvedic, yoga, and naturopathic, Unani, Siddha, and homeopathic. And early on, in the pandemic, there were, some claims made to different Ayurvedic and other products that these were really the cures for COVID. And eventually, I'm glad that a task force was set up and the ministry now really seeks scientific validation of the traditional medicines to make sure that they really are well vetted before they're recommended to the public. Next, please. And as of now, that I could see that there is no drug that has been approved for COVID. There are, various products that are in, various trials being tested. But I did see some good graphics, and they make some really, good general recommendations about remaining, hydrated and practicing yoga, pranayama, meditation. And I think that all of these are actually really good recommendations. And, so people, and I do believe that when people are rested and their minds are rested, their immune responses, work better as well. And I think some of these measures may have particularly a good role in the management of the post-COVID syndrome. Next, please. And I wanted to end with saying that how much I feel, really heartened at this huge triumph in India with the launching of this largest COVID-19 vaccination drive. And it makes me very proud. And this happened on January 16th. And, very much in Indian style, we have government officials praying over a box of vaccine doses in Mumbai. And really, we have 2 vaccines that are being distributed at this time, Covishield and Covaxin. Covaxin is manufactured and really kind of like an all India vaccine that the people and the scientists are very proud of. Next, please. And India is also setting a good example with what is vaccinemetry, really beginning to distribute millions of doses of vaccines to neighboring countries. And here is the Sri Lankan president receiving a box of COVID-19 vaccines from India. Next, please. And I wanted to leave you with some resources that I like to get to. There is the CDC resource, and there is the NIH resource that is, has some really nice tables and remains up to date about, various treatments that are recommended for different stages of disease. And Johns Hopkins, has a great website, and we saw Rayna use their dashboard, and it's kind of really nice to see, what's happening in different parts of the world in terms of the evolution of the epidemic. And others, New England Journal, Lancet have hubs devoted to coronaviruses, and those are free to access. And I really like to also use New York Times as a good resource. Thank you very much.
Well, thank you so much, Dr. Malhotra. I think what we'll do at this point is, let's see, I'm going to stop the share. And what I will actually do is, let me actually just move to these slides, if that's okay. And
thank you, Amish, for driving the presentation.
Yeah, absolutely. I'm not used to it very often, so hopefully
I haven't done that for a long time. So thank you for doing that. I really appreciate it.
Well, I wanted to definitely thank you for speaking, Dr. Malhotra, and I wanted to be able to invite Dr. Malhotra, Dr. Rayna Patel, Dr. Anita Chopra from the University of Washington, and Dr. Deepthi Mani from Multicare to join us to be able to answer questions from the audience. I know that many of you have sent questions through the chat box, and Dr. Trikudanathan is going to be checking those out and helping me today as we go through some of those questions. But there were a few questions I just wanted to throw out there to our members. And again, I would just ask for the audience, please don't unmute yourself to ask questions because it can be very disruptive. If you have a question, please send it through the chat box function. And we are following that, and we'll be able to respond to those questions. But I wanted to ask a few questions that have come up. One is, I just wanted to, again, ask, this has come up, and I think Dr. Chu helped to answer this a little bit, but the question of, based on what you have seen about the Pfizer and the Moderna vaccines here in the United States, do you believe that the vaccines are effective and will help us to reduce the transmission of COVID-19 infections? And part two of that is, do you anticipate that people will need a booster shot at some point, like a year from now, with these vaccines?
So I can take this question. I will say this, I guess, two parts to the question. One, are the vaccines efficacious? So I guess they're clearly, highly, highly efficacious. It's mind-blowing, I think, how efficacious they are. And the question is, the second part is, is transmissions, are they going to prevent transmissions? And we don't quite know that as yet. And there seems to be some hints that there is reduction in numbers of people who are asymptomatic. But the trials, there were no nasal swabs done in the trials, and the trials really looked at patients with symptoms and disease. And we know that asymptomatic patients can be a source for transmission. So clearly, I think I see the vaccines as really one layer of protection. And I think we need to continue with, using our masks and social distancing, um, and using the outdoors versus the indoors at this point. So to really, keep the uh, pandemic under control. And I'm sure that we will need boosters. I kind of see it more as us getting the influenza vaccine year after year. And at this point, we don't know the duration of immunity from from these vaccines. And I would have liked for it to be at least a year. I'm not sure of this at this point, given that we are, seeing this rapid spread of variants. And the part that I don't know is right now, most of the variants are just because RNA viruses really replicate and mutate, and there are spontaneous generation of variants. But what's going to happen when people actually start getting vaccines? So we may, start seeing more and more people. Right now, only small percentage of populations are vaccinated. But, once larger portions of the populations are vaccinated, we may see, specific variants that are really, specifically trying to escape the vaccine immune response. So we'll have to see whether we need, boosters even sooner. I think, which is why I think there's a great role for multiple vaccines um, in our toolbox.
Thank you, Dr. Malhotra. A question either, for another member of our panel. If you're an individual and you've received both of your COVID-19 vaccines and you want to travel to India to see family members, because you have to take care of people there because you have business to do there, would you feel comfortable saying that people can travel right now to India after being vaccinated? Maybe I'll throw that either to Dr. Chopra and then I'll let Dr. Patel then chime in.
All right. Yes, Rayna, please feel free to chime in. And from what we are seeing, I think that once you are vaccinated, there is still a chance, which of course we don't know about much, which is that you could still be an asymptomatic carrier even after you have received the vaccine, and you could still transmit vaccine to other people who are vulnerable. Of course, there is, all the infectious disease specialists and the public health specialists are still recommending non-pharmaceutical interventions, which is masking and social distancing. That goes without saying. Traveling to India would not be without a mask, would not be without, proper hand hygiene and protection. And of course, you would still, you would be protected, of course, if you were exposed to to be exposed to the virus. You would be, not have as much of impact of the infection, but you could serve as a carrier to someone else who is vulnerable. So one would still recommend, masking or social distancing when you reach there. And of course, the period of quarantine. I'm not very sure of, and I don't think there are any guidelines regarding the quarantine after vaccination yet. I think Rayna may want to pitch in if I'm not up to date on that. But I do believe that the quarantine times as far as travel are still in place.
Thank you, Anita. I'll just add 30 seconds. Indeed, this is the type of forward thinking that we're looking forward to. The reason we're all getting vaccinated is the hope of a new, better normal, including traveling. But at the moment, we don't have enough data yet to provide further guidance. So exactly as Dr. Anita Chopra said, no one has come up with guidance on travel after vaccination. At the moment, most countries are requiring testing before you get on the flight. And then in some countries, quarantine, a mandated 10 to 14-day quarantine afterwards. I do foresee things of that nature changing and morphing as more and more populations become vaccinated. But that's a bigger picture, intersection of policy and public health that is yet to be determined.
And then some questions that have come up about comfort level for certain things. For example, where are you with the comfort levels on eating out at restaurants, especially in the area of King County? And then comfort level about, when it's okay for kids to be able to go back to school. We're hearing about various school districts thinking about opening up in the near future.
So, I can speak here. And so, of course, children are, it's going to be a while before children get vaccinated. So most of the children will not be vaccinated. But we did hear from Dr. Helen Shu that, yeah, so children, the most majority of them get milder illnesses, and they are, there are, rare scenarios of severe disease. And so, but I think that it's sort of priorities. It seems to me like education here is a huge priority. So I would be more comfortable getting my child to school, and maybe, not as comfortable, eating in a restaurant. But it's again sort of priorities, where we, want to take the risk. And I think one thing to do kind of remember is that the vaccine, it's not 100% effective. It's the Moderna, Pfizer are like 94, 95% effective, and they're more effective in people who are younger. So if you are, an older individual, you may have maybe 85% efficacy. So sort of really kind of remembering that, and that, um, as you, make your decisions.
Dr. Malhotra, I might probably chime in to ask a question as a follow-up to this is, a lot of our patients who are on immunosuppressants or on chronic steroids who get the vaccine ask if they can find out if they've had immune response. So could you comment on testing about neutralizing antibodies, particularly in
Yeah.
clinic there?
Yeah, I think that's a really good question that comes up a lot. And unfortunately, our tests that are locally available, they are not neutralizing antibody tests. So neutralizing antibody tests really are, where you actually have the virus and you're trying to see, does this patient have, does their antibody really neutralize virus? And we don't really have, those capabilities. And we don't really have any data to actually say, what level of antibody is really correlating with protection at this point. So I think that the wise thing, is to just say that, that they, likely have received some protection, from the vaccine, but it's likely to be, a bit lower than what a young healthy person, would have had. So I think we can start loosening up, how we're living, but I think it's important for us to still continue to use, use protection. Like I feel more comfortable as I walking through the hospital, seeing my patients, even when I was masked before and still masked, I just have a different level of comfort. But I'm still, continuing to use all of those prior precautions. And same thing as I go to the grocery store and etc.
Subbu, if I may add
yeah.
a small component to that, is the Moderna and the Pfizer vaccine studies did include such populations in their trials. So cancer patients, I'm forgetting, one of them may have included solid organ transplant recipients as well, people living with HIV, not pregnant women. But so we have signals of efficacy in those individuals from their studies. They're not clearly powered to look at those subpopulations, but we have signals that suggest they should be efficacious in those individuals as well. So my personal practice, I recommend every single individual to get vaccinated. As Uma said, currently, I can speak for University of Washington laboratories, our COVID antibody testing is it's specific to like some of these spike proteins, et cetera. So it's not picking up the neutralizing antibodies that Uma Malhotra is talking about. So the flip side, the positive is, if you ever get tested for COVID, either by PCR or antibody testing, your vaccination will not interfere at the moment, the way our diagnostics are set up.
Well, thank you, Rayna. That was a very important point to distinguish. Yeah, thank you.
And then I have another question, and I'm going to direct this towards Dr. Mani, but a question from people about a lot of hospitals and clinics are limiting the number of people who can come in to the hospital or clinic right now. And also a lot of people expressing some concern that if they have family members who are admitted to the hospital, that they're having a hard time communicating with them. Do you have some thoughts as a hospitalist about what's going on here and how people can deal with this situation?
Hey, thank you, Amish. So it is correct that currently most hospitals have policies in place to limit visitors. I work at Valley Medical Center in Renton. And at Valley and most other hospitals, the only patients who are allowed visitors are the patients who are at end of life or comfort care measures only patients. In the emergency room, when they come in, we do allow visitors, but once they get transferred to the hospital ward, to the patient room, to minimize risk of potential transmission to others, we do limit the visitors. Having said that, most hospitals have in place various modalities like FaceTiming or Zoom calls,
Yeah.
which patients could utilize, which patients and their families could utilize to communicate with each other. We can also arrange Zoom calls with providers if that is what is helpful. We hope that once the vaccination rolls out and once more and more people among our community get immunized, things would change. But currently, when the levels of infection are still high, we are forced to put policies in place which limit the visitors. In certain circumstances, especially for patients who are demented or have a language barrier or they do make exceptions. So it's always still recommended that you ask the providers whether there could be an exception that is made, that they could be made on a case-by-case basis. Thank you.
Thank you. And another question, FDA coming in, if FDA approval of the current vaccines is only for emergency use because of lack of evidence for let me try again. If FDA approval for current vaccines is only for emergency use because of lack of evidence for long-term safety and efficacy, what duration of data is needed for approval of the vaccine for general use?
Good. That was my question.
Okay, I am happy to comment. Helen Shu is clearly our vaccine expert, so others can please chime in. So emergency use authorization does not imply lack of long-term efficacy or use data. It's the same data that the company will use eventually to have a full approval. What emergency use authorization implies is an expedited review process and a different type of authorization. So for those of you who received a COVID vaccine, you may have noticed how that administration is different than how you get your flu vaccine or other vaccines you've gotten. The difference is the systems that are administering the vaccine track very carefully what vaccines they're administering and how. And because of the emergency use authorization, we are doing, for example, a 15-minute monitoring of every individual who receives the vaccine afterwards. These all types of systems do not exist for normal vaccines that you get otherwise. So emergency use authorization does not imply lack of appropriate data. It's safe, it's efficacious, it's the exact same data that the companies are going to use for the formal approval. It's just that the process of approval was different, and that's why we have these slightly different processes to follow. I will say there's a slight difference between efficacy, which is the data that's observed in a constrained trial environment, versus effectiveness, which is what we observe when we roll out this vaccine in a real world setting. So yes, we are looking to receive effectiveness data, and some of that is beginning to be circulating now. But I imagine effectiveness data will be just as promising because the efficacy data is so amazing. We really, other than one other vaccine for HPV, we have no other vaccine that has this high efficacy data. This is amazing.
And then I'm going to direct this question, Dr. Malhotra, but could you speak a little bit more about the Regeneron monoclonal antibody and what we know about its effectiveness?
Yeah, so the monoclonal antibodies were designed to bind the spike protein. So which means that they are not able to bind the ACE receptor or the receptor on the human cells and cause an infection. So these antibodies were first trialed in patients who were hospitalized and actually did not show, any efficacy. And then, and it's kind of intuitive that the efficacy is probably in patients who are really early in the course of their infection, because we're still trying to bind the virus. And as we saw that by the time patients are admitted, majority of them are actually more in the hyperinflammatory phase, and there is no virus. So then the trials kind of focused on more people with early in the course of infection, and these are patients who are not hospitalized, just recently infected. And so as I recall, the benefit seems to be marginal, and which is why the focus has been more on folks that may be at really high risk. So current approval is people who are at really high risk for a complicated disease course, poorly controlled diabetes, high age, etc. And there are, I believe, very few hospitals in the city that are offering the antibodies at this point.
Thank you so much, Dr. Malhotra. A couple more questions. I'll do 3 more questions. There's a question about the use of aspirin to prevent strokes if you find out that you're COVID positive. I'm not sure who wants to answer that question.
I think it's a good idea.
Okay, and then there's a question about if you're a patient with cancer or with an autoimmune disease, what do you recommend in terms of the COVID-19 shots? And I'm happy to answer the autoimmune part. And I'll just say that the American College of Rheumatology is recommending that all patients with autoimmune disease get the COVID-19 shots. There's a couple of medications like methotrexate and rituximab. And if you're on those very specific medicines, you should touch base with your rheumatologist or the physician who's prescribing the immunomodulator therapies. There's a couple of medications for multiple sclerosis also that can reduce B cells also and make it harder to form an immune response. And so definitely if you're in doubt, as are many of my patients, touch base with your rheumatologist and send a question. They probably are able to answer it very quickly. And then I wasn't sure who wanted to touch base on the cancer question.
So, Amish, what's the question again about the cancer patients? Should they get a vaccine?
If you're being treated for cancer, should you get the vaccine?
Yes, definitely. And I think that the data have shown that active cancer, not cancer in remission, but active cancer is a risk factor for worsened disease course. And again, I think with both autoimmune diseases and different cancers, if there is a chance to be able to back off on the immunosuppression for a short period of time, before and after the vaccination, I think that's a good idea, just to allow your immune response to develop, to the vaccine.
And then one final question, which is kind of a bigger question, but people are curious about, there's obviously a lot of information coming out there about COVID-19. It's hard to keep up with all of the information. Where, maybe I'll just ask each of the panelists to just comment briefly on this themselves, but where are you getting your information from, or where are you directing your patients to get the most up-to-date information about, especially about the vaccines?
I think I'll start, and I think that UW has, the University of Washington has a great website which is accessible to everyone, to patients as well as the general public. And there is a page on the COVID-19 as well as a COVID vaccine. And I think they have some great FAQs that are out there, and people can look at that page. It also gives links to the Department of Health, the Washington Department of Health web pages, where you can actually find your phase and see if you are eligible for the vaccine as well. That is a great resource. King County Public Health has great resources for COVID-19, and so does the King County Medical Society. I looked at that page as well. I think for people in general, it's a good idea to know your phase, where you belong, and when are you eligible, and to have those phone numbers ready to know where the vaccines are available, what are the sites where they are available, and the phone numbers are listed on those websites as well. I think they are very handy for people. And then, of course, CDC is always the resource to go to. But I think all these websites, they are very well connected with the King County Public Health, the UW Medical Center, and the Washington Department of Health websites.
Hey, that was fantastic. Amish, I think most of the questions that came in the chat box got covered by Dr. Rayna Patel, Dr. Shu, and Dr. Deepti Mani. There were recurring themes, and if there are more comments that the panelists want to make, one of the questions that came was, what's the interchangeability between the Pfizer and Moderna vaccine, or between one platform and the other? So if there are any additional comments, that would be great.
No, I'll just say briefly, this is explicitly being studied in trials by the NIH. So in hopefully another 6 months, we'll have some answers for that.
Well, I wanted to just thank all of our panelists for taking their time. We know how busy they are as clinicians, and taking the time to talk to our community is so meaningful to us. And so on behalf of both the Indian American Community Services and WAPI, we're really honored to be able to organize this event. We could plan WAPI and Indian American Community Services to continue to have a partnership. We're planning 2 more virtual education sessions. On Saturday, May 8th, we'll be having a session focused on breast cancer, which is a cancer that disproportionately affects the South Asian population. And then on August 7th, we'll be having a discussion for the community about arthritis. We will be sending out reminders and emails as these come up, just so don't worry about having to write down these dates right now. But thank you again to all of our panelists for your time. We're really grateful.
Well, thank you, everyone. It's an honor and pleasure.
Thank you, everyone.
Yeah.
So exciting to see so many on today. Please reach out to each of us individually if you have any more questions.
Amish, thank you for doing such a great job with the whole platform here, and all the speakers were excellent. Nice to meet everybody, and we'll hopefully connect soon personally.
Yes, truly, congratulations to the organizers and Amish.
Well, thank you, everyone. This event is also thankfully to Lalita Pala and her wonderful team, her tech team, who are working around the clock at Indian American Community Services. This is being recorded, so a link to the YouTube version of this will be available in case you have family members or friends who couldn't be here today. We want to make sure that they'll be able to watch this at a different time. And certainly if you have family members who are vaccine skeptics, have them watch the video or reach out to us. We certainly want to get as close as possible to 100% vaccinations for all adults in our community as well. Thank you.
Yeah, thank you to everyone. This was wonderful, and we are looking forward to this partnership. Thank you.
Bye-bye.
Bye to everybody. Lalita, thank you.
Thank you.
Amish, wonderful.
Oh, thank you, Tilaka, for tuning in. We really appreciate it.
Of course. I think it was really successful. We had, what, 78 at all times.
Yeah, absolutely. And I think hopefully more people will watch this at a future date as well. Yeah.
Yeah, we streamed it on IAWW pages.
Thank you so much for streaming it. I really appreciate it.
Of course. Thank you.
Okay. All right, I will step out too.
Bye-bye.
Thank you, Amish, and we will email you the YouTube link.
Thank you so much.
As well as the raw footage. Bye-bye, everyone. I'm going to end this now.
Okay.